We've looked at transcription and translation, and the reason that those two processes are so important is because they are making proteins, and making a protein is important because your body has to make many proteins. It's not just protein that you eat through your diet, but it makes many of its own proteins from your DNA. All of the traits that you have that we talked about in genetics, like your eye color and your hair color and all those things, are determined by proteins. So when we make a protein, it is important, and proteins can also have mistakes in them, and so we're going to look at mutations for these last two videos to finish up our notes. A mutation is any change in your genetic material. Whether the change leads to hurting the protein, sometimes a change doesn't affect the protein at all, it's still considered a mutation if there's any change to your genetic material, which would be your DNA. In the video and also in class, you're going to be hearing me use the word gene mutation, but you'll also hear point mutation, and those are referring to the same thing. So I just didn't want you to get confused. When you see point mutation or gene mutation, we're referring to any change, and usually it's one base. Sometimes it's one or two, but it's a very small number of changes to your DNA. And when we say a base, we're referring to the nitrogen bases of adenine, guanine, cytosine, and thymine, which is one of those parts to a nucleotide. Now, if the mutation occurs in your gametes, which those are your sex cells, if the mutation occurs in your gametes, then that gamete that has the mutation in it could be the gamete that gets passed on to the offspring. So there are times where the mutation can get transferred to your children, and if the mutation affects your ultimate phenotype, then we would call that a genetic disorder. So we do have multiple genetic disorders that you'll be hearing me refer to in class and through some activities that we do. So let's look at some types of point mutations, and there are three types. We're going to look at each one on its own separate slide. But the three main point mutations are substitutions, insertions, and deletions. Now if we look at a substitution, you're going to notice that it is just replacing one nucleotide for another. So it wasn't proofread correctly. We have the wrong letter there from what it should be. So all you're doing is substituting one base, one A, G, C, or T for another. And the example that I'm going to look at is an RNA strand. So this would be what the original RNA should look like when it was decoded from the DNA, but the mutation is going to happen right here. So notice all the letters are the same except for this one, and all we've done is we've substituted, instead of a C being here, it's an A. So when it was going through transcription and it was making the messenger RNA copy, instead of the G going to a C like it should have, the original G from your DNA paired with an A, which shouldn't happen, and that's why this is called a mutation. Now there are a few different things that can happen when you have a substitution mutation. First of all, sometimes you could get the same amino acid and the protein isn't going to change at all. So that is a possibility. And if we take a look at this example, it's important to look at what we should have. So this is what we should have right up here: methionine, lysine, phenylalanine, glycine, and then a stop. So just if we're looking at those sequences, we're going to take a look at this top example right here. This GGC, and if you have your circle chart out, you can look up GGC and notice that it was supposed to be glycine. But the mistake right here, we now have a U. So we've substituted, instead of a C, we have a U. And so if you look up GGU on your circle chart, you're going to notice that that's also glycine. And so it's the exact same amino acid, even though it's still called a mutation because we have a mistake from what the original should be. It didn't affect the protein or the amino acid sequence. So that's a possibility that can happen. We also have times where a substitution can give us the wrong amino acid, and when we have the wrong amino acid, then our protein has a mistake in it, so we have the wrong protein. And if we take a look at this example right here, instead of us having a G here, we have a T. And so once we have a mistake in our DNA, it kind of domino effects to the rest of the process. So when DNA goes through transcription and we get our messenger RNA, this G should have been a C here. But because we have the mutation, we have a T, T pairs with A. And so if you look at GCU on your circle chart, you get proline, but now we don't have CCU, we have ACU, and that gives us a completely different amino acid. So now our protein is different since the amino acid sequence is wrong. This happens in sickle cell disease. Sickle cell is where the hemoglobin in your red blood cells is deformed, so it doesn't work properly, which can be incredibly dangerous because hemoglobin is what carries oxygen in your red blood cells. So if that hemoglobin is deformed, it's not carrying the oxygen the way it's supposed to, and that can cause a lot of organ damage, possibly even organ failure, and it also creates a lack of energy because oxygen is what you need to make energy. So sickle cell works this way. There is a one-letter difference between a normal hemoglobin protein and the protein for hemoglobin that's sickle cell. One substitution mistake, excuse me. And this is what the hemoglobin particular spot is. We should have this CTT on normal DNA, so most of us would have CTT, and that on the mRNA, C goes to G, T's go to A's, and GAA is, I think it's glutamic acid, GLU. Now if you look at a person with hemoglobin, instead of the original CTT, they have CAT, C-A-T. One letter difference, but that one letter makes the RNA also incorrect, and it, in this case, creates a different amino acid valine, and that valine, even though it's only one amino acid out of like 400 that hemoglobin has, creates the deformed shape. So one letter can make a huge difference in the life and the health of that individual. Now, we need to look at frameshift mutations, and we mentioned this in an earlier video. Frameshift mutations are going to affect your reading frame. Remember, we read these in 3-letter codes, and so the number of nucleotides are not going to be in multiples of 3 in a frameshift. We are going to end up changing quite a few amino acids. So wherever our shift happens, we could insert a letter, which shifts all of our letters over. You could delete a letter, which shifts all of your letters back a space. Either way, you're shifting the reading frame, and so you are going to be affecting multiple amino acids. Wherever the mistake is, every amino acid after that could be affected. This one, these frameshift mutations can actually alter your protein so much that it can't perform its job at all. And so you might have like a deficiency with that particular protein just because it's so altered and so changed, and proteins proteins do their job based on their shape. So if the shape is wrong, then the protein can't work or can't work correctly. The examples of frameshift mutations would be the insertion and the deletion. These are point mutations because they're affecting one letter, but yet they're also frameshift mutations because they're going to be moving the other DNA nucleotide letters around. So let's just look at an insertion. This is where you add a nucleotide. So if you take a look at what your normal DNA should have been, and then this would be the ultimate, your mRNAs and your amino acids. So we have transcription and translation here. All we've done is in this spot, and they have this arrow between the T and the C, they've added a letter. So all the letters are still there, GAT, all of these letters here, But since you've inserted a letter, remember we read these by threes, so every 3-letter codes for an amino acid. And if you group these in threes, you can see that all of our 3-letter words now are off a letter. So your amino acid from the point of the mistake and everyone after that, these are all wrong. These amino acids are from what the original should be. Now before the mistake, this leucine amino acid still the same in the bad one. So wherever the mistake is, everything before that's okay, but everything after that is going to have some major mistakes. So you notice every amino acid after the point of insertion is wrong. So the more mistakes in amino acids you have, then the more that protein doesn't function properly. And then deletion, very similar, only you're deleting a letter, you're losing a nucleotide. So in this one we're taking this G away, so that shifts all of our letters over a spot, and so instead of GAA giving us the glutamic acid or amino acid, we now have AAG, because they're still going to get read in 3-letter codes. So since all these letters got shifted over a space, that gave us AAG, and on our circle chart that is a different amino acid. So again, from the point of the mistake and on, all of these amino acids are going to be incorrect. And remember, you do need 3 letters, so in this case you get to the end, you don't even have 3 letters anymore, so there's no even stop codon here, so your body's going to be, really can't even make the protein since there is nothing to tell it to stop making it. So those are some gene mutations, point mutations, and we'll look at some more mutations in a later video, but guaranteed that these 3 are definite ones you need to know. And you need to know that frameshift mutations like insertion and deletion actually cause more damage to the protein because of the number of amino acids being affected.